So we have a system that's akin to Roundup Ready corn, where suddenly we have like a herbicide, and then we have a plant that is genetically different from surrounding plants that is resistant to the herbicide, but the surrounding plants are not.
We have, a naturally occurring organic peptide hormone herbicide, of which there's nothing like it really yet.
There's an open market right there, right? Like, we need an herbicide that is peptide based, and so I immediately knew that this would be a great system. It's about being more eco-friendly and natural, right? Because it's an organic system, and also it is just in general better for crop production. As we see increase in temperature in some areas and a decrease in rainfall, then we're seeing an increase in drought, and these warmer conditions and less wet conditions prevent the traditional herbicides from breaking down. They can leach into our water supplies, and that has the potential to be harmful.
Virtually all insecticides, fungicides, herbicides that are currently on the market, aside from really quite a small handful, are small molecule based, and as we've learned time and time again over the past few decades, small molecules don't like to go away, they don't like to break down naturally, and they have side effects that we can't predict.
But beyond that, there's this thing called crop rotations, where you will plant one crop, and after you've grown that crop and completed that harvest, you then switch to a different crop. Some crops are going to be resistant or hardy against some herbicides, while others will not be. And so you might have to increase the period in which you have to wait before you can start that second crop due to the lingering herbicide. It's the silliest story, because I was just waiting for the bus, trying to think of how I could spin my research, kind of spin it so that I could apply for my next grant. And I had this realization that a great way to make my research interesting would be to explore how it's very obvious that once a plant is subjected to high enough amounts of the peptide that I work on, it causes the leaves to yellow, which indicate that the plant's dying. It's called SPH. It just stands for S-protein homolog. It is only expressed in reproductive tissues, which is why I'm interested in it, because I'm a plant reproductive biologist. Naturally, it's involved in flower morphology, so the way the flower looks.
Paige came to our lab a few years ago. She was interested in looking for the receptor for this hormone that she was working on, and Mike and I sort of brought our background to bear on that, and so ultimately, we discovered what is likely one of the receptors for this peptide hormone. Knocking that receptor out in plants that we care about leads to insensitivity, means that we could blanket apply this naturally occurring peptide hormone to fields of crops that have this one small knockout. Weeds would be killed, other invasive species could be killed, and the crops themselves would nonetheless be fine. So there's a lot of potential for this discovery, this peptide hormone pairing specifically, that we've discovered to have commercial impact.
These peptides are just going to break down and dissolve and not be an issue in terms of crop rotations. They're not going to leach into our water supplies or anything along those lines. I have shown that it is applicable to other plants outside of Arabidopsis, the model organism. Our future directions, so the next steps that we would like to take, would be to expand our exploration of the phenotype. So if we take that peptide and we treat other things like different weeds or different crops, if we also see the same phenotype, because that would mean that it's more applicable.
There's a lot of things we could do, but from a translational or application standpoint, testing it in other systems, other plant-based systems, scaling up and producing it in less expensive ways, and learning as much as we can mechanistically about how these things interact so that we might develop iterative and or better versions of this for future use.
Every crop that I've looked at the genome of has both the peptide, sequences for the peptide, and sequences for the receptor, right? So that means that the system should work in them regardless. It's just a matter of actually doing the empirical work.
Hi, I'm Jennifer Gottwald, Director of Licensing at Wharf. To learn more about this technology, please use the contact information on the screen. And please remember to like and subscribe to see more of our great videos.